All right, guys. So at this point, you should have finished working on the part 3 from the What Do Enzymes Do from yesterday and the Exploring Enzymes page that you picked up when you came into class. We're going to take a little bit of time and go over part 3 and talk a little bit more about enzymes before we move into talking about the cell. So let's look at part 3. Part 3 in this activity gave us data on lactase activity in different environments, and we're going to look at how those changes in environments affected how well lactase worked on lactose to produce glucose. So first off, the data was kind of confusing for a lot of you, so let's look at it. First off, the milk was designed only as a control. And we tested this to show that before we started, there was no glucose in the milk. We did that just to show that there's no glucose, because if there had been glucose, then the rest of our readings wouldn't have meant much. But since there's no glucose, we know that any glucose we get is because lactase has been breaking down the lactose. So our first graph, they ask us to graph how pH affects lactase activity. So we want to talk about pH. So we know that this information, this data for pH is important, and lactase activity data is important. So we know that those are the two columns we're going to look at. Then we say, okay, well, what data are we going to actually graph? So let's look at it where it says milk only, and it's a control, and it says do not graph. That means this will not be part of our graph. So now we say, okay, well, what data are we going to graph that involves pH and lactase activity? So this is what we have right here. And we are going to create a line graph to show the change in activity as we change the pH. Now, line graphs usually show change over time, but it can also show change over a range. And pH is a range. pH ranges from a pH of 1 to a pH of 14, where 7 is considered neutral. 14 is highly basic or alkaline, and 1 is highly acidic. Our stomach is at about a 1.5. We see salsa and citric acids. We see that about a 3. Bleach is over here at about a 12 to 13. And bases and acids are both highly caustic. The further you get away from neutral, the more that caustic they are. Coke falls in right about a 2. And milk is a 6. So what we're going to do is we're going to look at pH and lactase activity, and we're going to see how the activity changes over the range in the pH scale. So pH will go on our x-axis, lactase activity will go on our y-axis, and we don't want to forget to include our units of milligrams per deciliter. And then we're going to figure out what scale we should use. I'm thinking if you count by 2s, you should be able to fit the scale all the way to 14. If you use a different scale, as long as it fits and it goes through, uses most of the graph, you're fine. For lactase activity, we have to get from 0 to 1,000. So I'm going to mark every other line, counting by 2s, or counting by 200. If you use a different scale, perfectly fine, as long as it's even throughout. And then we're going to graph our data. So the control has a pH of 6 and a lactase activity of 1,000. So if we go to a pH of 6 and 1,000, we're going to put our dot there. And then the pH of 1 has a lactase activity of none. And then a pH of 4 has a lactase activity of 300. And a pH of 12 has a lactase activity of 0. Once I have those plotted, I'm going to connect the dots. And yours will probably look a lot better than mine on this board, but you should have a peak like this. Now, what does this peak show us? It shows us that lactase works best at a pH of 6. That's where it has the most activity. And as we leave the pH of 6 and we get further to the acidic side or further to the basic side, we see less and less activity. This tells us that lactase has an optimal pH range, and the further we get out of that range, the less it works. Now remember, lactase is an enzyme, and we said that enzymes have a very specific shape, and the shape of their active site allows for their substrate to attach to it. We also talked about how changes in the environment can cause the shape of the enzyme to change. And if the shape of the enzyme changes, it denatures and it no longer works. pH was one of those environmental conditions. So in the optimal range, lactase is working great. But once we get out of that optimal range, we see that the lactase stops working. And since we understand that pH can cause denaturing, we know that as it gets further and further out of that optimal range, the more and more enzymes are becoming denatured. And when we get to a pH of 1 and a pH of 12, there are no functional lactase enzymes anymore. They are not catalyzing the reaction, and we are not getting any glucose. So that is the graph and what we could get from the graph. So that should answer questions 9 about the observations for pH. Now, question 10 asks us to make a general claim about the effects of pH on enzymes. Well, what we do know is that lactase works best at a pH of 6, but there are other enzymes like pepsin, and we might find pepsin, or we find pepsin in our stomach. And in our stomach is extremely acidic. It's a 1.5. So pepsin is going to work best at a 1.5 because that's where it is. So if pepsin works best at a 1.5, how do you think it might work when it's at a pH of 6? It's going to work very similarly to the way lactase worked outside of its range. So if we get here pepsin at a 6, we might see it working about half of what it was doing before. We're going to see a drastic drop as we leave that range. The point being, enzymes have a very specific pH, and the pH that they work in is the pH that they are designed to work in the environment where we find that enzyme. So enzymes in the stomach work better in a range that's more acidic. Enzymes in our blood work better at a more neutral range. So every enzyme has a very specific pH range in which they work, and outside of that range, they denature and they no longer function. That is how we would answer question 10. And then we would cite specific evidence from the graph about lactase to support that claim. Lactase has an optimal pH at 6. As we move further and further away from the pH of 6, we see the activity of lactase decrease. So let's look at the next graph. Now we want to talk about temperature. And so on this one, we want to look at the data table and say, okay, what data are we going to use? We're looking at the effect of lactase activity on temperature. So we know we want to use temperature, and we know we want to use lactase activity. We know that milk by itself was used as a control to prove that there was no glucose, but it says do not graph it. So we're going to mark that one out. We're not worried about pH, so we're going to look at where we have data for temperature. Temperature is our independent variable, so we'll put temperature on the x-axis, and we're going to put our units of degrees Celsius. On the y-axis, we're going to put lactase activity. And we're going to put our units again, so check yours and make sure that you have your units posted. For our temperature scale, we need to go from 20 to 37. So if we start with this being 0 and we count by 20s, by 20 degrees at a time, we should be able to fit the scale pretty well. And then for our lactase activity, once again, I'm going to do every other line, marking it by 200s. And then I'm going to look at my data and say, okay, Where does it say? Where do I plot my points? So 37 degrees, I'm going to be at 1,000, so about there. And then at 20 degrees, I'm going to be at 300. And at 100 degrees, I will be at 0. And so we get a graph that looks something like that. Once again, we're seeing a peak. That peak tells us that lactase has an optimal condition that it works in. And that optimal condition, that optimal temperature here is 37 degrees Celsius, which is about equivalent to 98.6 degrees Fahrenheit. So body temperature, lactase works best at body temperature. As we move further and further away from that body temperature, that optimal temperature range, we see less and less activity. Now remember, as molecules get colder, they slow down. It doesn't necessarily denature the protein, but it causes them to slow down. And when they slow down, they have less of a chance of the enzymes running into the substrate. So we have less and less reactions occurring. But as we heat it up and we get hotter and hotter and hotter and further away from a functional range, we start to see the enzymes change shape. The bonds in those molecules in the amino acids that make the enzyme change, and they change shape. And when they change shape, we call it denatured. And when it's denatured, it no longer works, because the substrate can no longer fit into the active site. So here we see denaturing happen. And as it denatures, we see less and less activity. On this side, we see the decrease because the molecules are slowing down. Here is where we see the denaturing. Now, lactase is an enzyme that we find in our body, so it makes sense that it works at body temperature. Now, in bacterial cells that live in the deep, dark depths of the ocean and live in the thermal vents, they function at a much different temperature than we do. But they are living cells, and they do have proteins, and those proteins do catalyze reactions. But those enzymes are going to work at a different temperature than ours. They're going to work at a much hotter temperature when they're in those thermal vents. So their optimal temperature range is going to be different than ours. If we were to plot their activity, they would have a peak in their optimal range, and we would see a decline outside of that range. So for question number 11, based on the data, write a general claim about how temperature affects the activity of most enzymes. Enzymes have an optimal temperature range. Outside of that range, we see a decrease in activity. When it gets too cold, the molecules slow down, and they slow down, and they are less likely to collide with the substrate, decreasing activity levels. When the temperature gets too high, it causes denaturation of the enzyme, and substrate can no longer fit. Therefore, we have no more reactions, and the rate of reaction decreases. All right, so I hope that kind of helped you understand a little bit more of what you're looking at with those enzymes. Now we're going to look at the Exploring Enzymes worksheet, and you had to read the first few paragraphs, and it talked about catalase, and it talked about how catalase is an enzyme that can break down hydrogen peroxide into water and oxygen. And catalase, we find it present in our cells. They're in plant cells, it's in animal cells, there's a high amount in white blood cells. And catalase is used to help break down toxins, and hydrogen peroxide is a toxin. So if hydrogen peroxide were to be produced in our cells, catalase would break it down quickly, so our cells would not be in trouble. When we use hydrogen peroxide to pour on a cut, we're pouring it on a cut, and if it bubbles, it means there are some cells that are open, whether they're white blood cells or other body cells, but it breaks the hydrogen peroxide down into oxygen and into water. The oxygen is what creates the bubbles. Water helps flush out the cut, so that's why we use hydrogen peroxide on cuts. It does help flush it out. So once we finished our reading, it asked us in question number 1 to label this diagram, and it asked us to label the catalase. Remember, ASE tells us it's an enzyme, and the enzyme is the holder. It holds the substrate. So if you look at the picture, this would be our catalase. And remember, our catalase stays the same throughout the reaction. Enzymes are not changed or used up during a reaction. And catalase has a very specific site. It is called an active site. And the active site is where the substrate attaches, and my substrate got cut off here. But the substrate in this reaction is the hydrogen peroxide. When the substrate attaches to the enzyme, this arrow represents the substrate binding, or you could have labeled it here and said substrate binding. What is released from the enzyme at the end of the reaction we call our products. And in this case, it would be oxygen and water. And when this substrate is actually attached here to the enzyme, we give it a special name. This is a review from yesterday. I didn't ask you to label it, but I just want to review it with you again. That together is called the enzyme substrate complex. Then on question number 2, it asked us to look at this graph and analyze it and go and label different parts of it. So to do this, we had to remember that an enzyme speeds up a reaction, and the way it speeds it up is by reducing the amount of activation energy, or the energy needed to start the reaction. It's the energy that has to be invested to make the reaction start. So we look at this graph and we say, okay, we have the reactants here. And in order to get to the products, we have to invest. Our y-axis has energy. We have to invest energy to get to the top of the hill in order for us to get to the product. So we're going to invest some energy into it. That is our activation energy. So our reactants here, we invest a bunch of energy to get to the products. And then in this blue line, we invest a lot less energy. So the one with less energy, this line, would represent the reaction with an enzyme. Because remember, enzymes reduce the amount of activation energy required. So this piece would represent the reaction without an enzyme. So now these blue arrows are talking about the measure of energy from the reactants to the top of the peak. So this blue arrow would represent the activation energy without the enzyme. And notice it is much larger than the activation energy with an enzyme. Because it takes less energy to invest to start the reaction, it's going to take a lot less time. We're still going to end with the same products either way. But it takes a lot less time. It's much more functional and much more efficient for living organisms to use enzymes in their reactions. And remember, enzymes are used in just about every single reaction that we have. So why did we start this unit with enzymes? This is the cell unit. Well, we started it because remember, enzymes are proteins. Where do proteins get made? If your organelle is a ribosome, you know the answer to this question. The ribosome is what makes proteins. Well, so ribosome is an organelle in a cell, so cells make proteins. Those proteins can be enzymes, but also enzymes catalyze every single reaction in a living organism. So most chemical reactions occur within the cells. So those cells, all those processes that you're learning about as you learn your organelle, cellular respiration, photosynthesis, protein synthesis, DNA replication, mitosis, all of those things are catalyzed by enzymes. So we had to understand what an enzyme does so we can understand how things are happening in the cell. So that's how we started. Now we're going to jump into the cell and start learning about various organelles in the cell and various processes in the cell. So we're going to see what you know about the cell. So right now I want you to complete the recalling prior knowledge of cells activity. For those of you at home, it is linked on Google Classroom. You can print it out or answer it on notebook paper, but there are 3 questions, a little paragraph to read. And then on the back, I want you to identify as many organelles as you can. The sheet gives you the descriptions. Write the names of the organelles that you know as you read those descriptions. If you don't know it, leave it blank. You can take your best guess, but I do recommend using pencil. We're not going to get the correct answers today. I want you to see what you know now, and then I want you to see what you know after we learn the organelles and go through them tomorrow when we watch our videos. So you can go ahead and start that right now. It shouldn't take the full 10 minutes, but I'm going to go ahead and start the timer for you. All right, guys.
Speaker mode.
All right, guys, let's go ahead and talk about the first 3 questions. What functions must cells perform? I was walking around and saw several things that y'all wrote. You wrote mitosis. If you don't have these, you can go ahead and add them to it. Photosynthesis. Cellular respiration. Some of you wrote that things enter and leave the cell. Some of you wrote break down food. Build lipids. Protein synthesis. Guys, this list could go on and on and on and on. If you think about your organelle, the function of your organelle, that is the function that the cell has to perform. This list could go on forever, but what we need to know is that the cells can perform these functions, but how do they perform those functions? Each individual organelle has its own function, and all of those organelles function together for one common purpose, for the functioning of that cell. So how is a cell like a system? Well, remember our body systems. When we talk about a body system, the circulatory system, the respiratory system, the system is a bunch of organs that work together to serve a purpose within an organism. Well, a cell is just like that. A cell is a system. It has little organelles. Organelle literally means little organs. So like our systems have organs, the cell has little organs or organelles. And each of those organelles perform specific jobs that allow the cell to function as a whole. So how is a cell like a system? Each organelle functions for a specific purpose allowing the cell to function as a whole. And what does it mean that the form of a cell structure often predicts their function? Well, we kind of skew your thoughts a little bit when we teach you about cells in elementary and middle school, because we show you a diagram and the plant cell is a square, which a lot of plant cells are, but not all of them are. And the animal cells are spheres or circles, and they're not all spheres or circles. Our nerve cells are really long, and they extend a long way, and then they have these branches that reach out. And why do they have that? So they can transmit a signal. Our skin cells form sheets or layers of skin, and the skin cells are more square. They're not plant cells, they're animal cells, but they're more square. But there's the way their shape allows them to line up right next to each other and create this solid sheet that we see as our skin. So the shape of the cell helps determine its function. The way it is designed, it's designed to for the function that it serves. Nerve cells are really long, so they can send that signal really quick down your arm, through your body, and your body can respond quickly. So that's what we're talking about when it means that the form of a cell's structures often predicts their function. And so let's look, on the back, you looked at organelles and asked you to identify which ones you thought they were. I am not going to give you the right answers right now, so just hang on to this. But know that this back page, well, this whole page is a great study guide. So on the top of it, if you want to write awesome study guide. Write that up at the top of the paper so that you know to study this. You're going to have a quiz next Tuesday, and a big part of it is going to come from this paper. So make sure that you keep up with it, and tomorrow after we watch our videos on Flipgrid, we'll go back and see if you can figure out the right answers. You will have the correct answers to the organelle descriptions before you take the quiz, but right now I just wanted you to see what you knew, and then you're going to go back and reassess what you know after we talk about the organelles tomorrow. So that was our pretest. So what we're going to do now is you are going to work on the prokaryote and eukaryote POGL. For those of you at home, it is attached to the Google Classroom post. You can print it out. It does not take very long. It is not super hard, and you don't have to know anything in advance to do it. Use the diagrams to answer the questions. When you finish, you're going to work till the bell, and when you finish, you will just hold on to it. Tomorrow we will finish that. We'll finish it up if you don't have enough time today, and we'll create the Venn diagrams tomorrow before we watch our videos. A few reminders for tomorrow that your organelle video, if you're at home, this goes for you as well. Your organelle video must be posted by 11:59 p.m. tonight. And your cardstock profile is due tomorrow. Now, if you're at home, just bring me your profile whenever you're back in class. But make sure that that cardstock profile, there's a picture of it or it's in your video for at least 20 seconds. There are some people who are taking a picture of it, and you can upload the picture to your video on Flipgrid, and they're making a split screen to where the profile covers one half of the screen and your face is in the other. However you decide to do your video, but make sure that profile is in there for at least 20 seconds and your video is 1 minute to 3 minutes long. Post it before 11:59. Get that done as soon as you can. So if you have issues, you can send me an email and we can get it figured out. If you post it at 11:58 and your Wi-Fi goes out, there's not much I can do for you at that point. You've had this assignment for a while. Animal Behavior Lab is also due tomorrow. Please make sure you have that to turn in. And one more thing, please make sure you bring Chromebook compatible headphones tomorrow. That means if they plug into your iPhone in that lightning charger spot, they will not fit in a Chromebook. Chromebooks will Bluetooth to Bluetooth headphones, but please bring your own. I have some I can lend out, but I would prefer not to have to deal with trying to disinfect those if I don't have to. If you own your own, it's probably more sanitary for you to use those, so just make sure you have them with you tomorrow. All right, guys, so y'all work on your prokaryotic, eukaryotic POGL until the bell rings, and y'all have a great afternoon.